Beginning chapter 16 on blood today. Not as long as some of the other chapters that we have, but nevertheless important, and probably we don't spend enough time understanding the role of blood in basic physiology. So that's the goal of including this chapter. So about this chapter, things we're going to cover, we're going to go through some of the, shall we say, anatomy of blood, the plasma and the cellular elements of blood, how blood cells are produced, a process known as hematopoiesis. We're going to talk about red blood cells in particular. I will get into some white blood cells, but really not a whole lot. That's more in the immunology side of the equation. And so we're going to focus a little bit more on red blood cells than the others. And then finally, in terms of mechanistic content, we're going to talk about platelets and the process of what's called hemostasis, or if you're from the UK, hemostasis. In any case, that's about coagulation. Let's get into the quote-unquote anatomy of blood plasma. Yeah, sure, the cells are very, very important. Red blood cells, important for life, white blood cells, immunity, and so forth. But we also need to think about the proteins as well. There's a protein anatomy in the blood plasma. And we have four major groups. I wouldn't call them families. The first two are families. The second two are just individuals. In any case, these guys comprise the majority of proteins in the blood plasma, but not the totality. Let's start with the first group here, albumins. These guys, their function is that they provide a huge percentage of the osmotic pressure in plasma. You remember back in chapter 15, we had hydrostatic pressure pushing fluid out of the capillaries, and we had the letter pi, which stood for colloid osmotic pressure, pulling fluid back into the capillaries. Well, albumins do most of the workload for that. They are the major protein in blood plasma. So albumins are a huge contributor to colloid osmotic pressure. And secondly, they are carrier proteins. They have a hydrophobic interior that allows things that would normally not be water soluble to move through the bloodstream. And that includes things like steroid hormones, testosterone, estrogen, androstenedione, all of those things, cortisol, are all examples of things that are carried by albumins. One other thing I'd like you to notice here is, and I should have said this a second ago, but most of these proteins are manufactured by the liver. And so if you have liver disease, one of the first manifestations that you see is edema and other things associated with deficit of protein in the blood plasma. So we've got albumins done here. Okay, they are colloid osmotic. Okay, they pull fluid back into the blood plasma. They're also carrier proteins. Now let's move to the next family, the globulins. And these guys a lot of them are potentially enzymatically active. Globulins are, by their shape, they have a kind of a bulbous appearance if you look at them. Again, made by the liver. A lot of our clotting factors are globulins. Enzymes present in the blood plasma are globulins. Antibodies, in fact, we call antibodies like IgA or IgD or IgG. That Ig at the beginning stands for immunoglobulin. Okay. Carrier proteins as well, but they're primarily, if I were to say one thing, is that they retain enzymatic activity. Even antibodies have a little bit of enzymatic activity. So these guys are the metabolically active proteins in blood plasma. We have the precursor to a clot, a true clot, a precursor to true clot called fibrinogen. And if you see ogen, it means that it is a precursor of something. You see that suffix on the end of a word, that means precursor. Fibrinogen, plasminogen, and so forth. Precursor to fibrin. What happens is fibrinogen gets cleaved by a clotting factor, and it starts polymerizing into fibrin. And that will trap red blood cells, white blood cells, and create a true clot. We have two different kinds of clot. We have a loose platelet plug, and we have a fibrin clot. This is the latter. And then lastly, an iron-carrying protein, again made by the liver, iron-carrying protein called transferrin. Transferrin keeps iron under lock and key. any of the bivalent metals, okay, zinc, calcium, not so much in the blood plasma, but we'll leave that one there. Iron, copper are kept under lock and key, especially iron and copper. Okay, they're very capable of creating free radicals, and so we have to have carrier proteins to prevent those ions from catalyzing the creation of free radicals. And so we transport iron using a protein called transferrin. We're going to come back to transferrin a little bit later, but you should be able to recognize these. You should be able to understand a basic function, if you will, of each, and you can tell that most of these things come from the liver, which makes the liver ever so more important. On the cellular element side of the equation, we have three major ones / groups. The big one, the one that is the majority of cellular elements, would be called erythrocytes, the red blood cells. And these are basically carrier elements for oxygen. That's their primary role. There's some other ones. I'm being a little glib there. But the erythrocytes are involved in oxygen transport. We have platelets. Platelets are actually subcellular fragments made from cells known as megakaryocytes. Sometimes we refer to platelets as thrombocytes because they are involved in the creation of a thrombus, which is a clot, usually a clot in place. Now, if that thrombus travels, then we start calling it an embolus. So if you've ever heard of a pulmonary embolism, that's a clot that's traveled from its original location. We could also say any type of occlusion, like for instance, a bubble of gas could be considered an embolus. In any case, our third group are white blood cells, and there's a whole bunch of those. So there's a whole bunch of different types of leukocytes, each with relatively discrete but interweaving and overlapping function. So here are our erythrocytes on the left, here's our platelets, and then over here we have all of the white blood cells, if you will, the leukocytes. There are also lymphocytes out there, but they sort of lump into the leukocytes. So different types of leukocytes, lymphocytes, neutrophils, eosinophils, basophils, they all have discrete but overlapping and synergistic immune and anti-pathogenic function. Okay. For instance, lymphocytes are involved in what we call adaptive immunity, and that's a big deal right now. Adaptive immunity is what we develop in response to some type of viral infection. You have to be exposed, or somebody has to be exposed for antibodies to be generated. That's what we mean by adaptive. It requires a cell to generate those antibodies and cell-mediated. Neutrophils are the sort of the big workhorse when it comes to basic inflammatory responses, and particularly bacterial infections. So neutrophils are involved in inflammation and bacterial infections. They're sort of the front line responders to an infection. Eosinophils we often see with allergies. We see them in parasitic infections, and like neutrophils, they overlap a little bit with inflammation. In fact, certain forms of asthma are associated with eosinophils. The really sort of nasty refractory asthma, the one that doesn't respond to normal therapies, is considered to be what's called eosinophilic asthma. But you can think in terms of function, parasitic reactions in particular, allergic reactions. Basophils are big time for inflammation, non-specific immunity. They are full of granules called histamine, and they're involved in the inflammatory signs and symptoms that you associate with inflammation. That is swelling, redness, heat, so forth. That's the role of histamine, pain. That's the role of the basophil. Now, I'm not going to get into hematopoiesis per se, with the one big exception here. The one big exception is that I want you to understand that we start with what we call a pluripotent. That is to say, this stem cell, this pluripotent hematopoietic stem cell, can differentiate into every type of blood cell. So it can form all of the blood cells, and that's why it's pluripotent. Okay, and as it differentiates, it's committed to a fate. And how do we determine that fate? Cytokines. Cytokines, cytokines, cytokines drive the differentiation of these stem cells into their specialized cell forms. So it's cytokine driven, and we know this now because we can actually artificially grow these stem cells, and we can create certain types of cells based on the cytokines that we give those cells. For instance, if we give the cytokine erythropoietin, we will cause red blood cells to form. If we use other ones, we will cause other cell types to form. And I'm going to give you a couple of those. Okay, so the differentiation from stem cell to differentiated cell is almost entirely cytokine driven. And this happens in the bone marrow to a large extent, that these cells, the stem cells are found in the bone marrow. Interestingly, though, we find a high population of these stem cells in umbilical cords. So at the time a baby is born, there are a high percentage of stem cells in that umbilical cord blood, and they use those from time to time. But most of our cells arise from the bone marrow in our long bones. And they will be exposed to cytokines, they'll differentiate, and they'll migrate and eventually move into the bloodstream. Some things like lymphocytes will partially differentiate in the red bone marrow and then move to lymphoid organs, lymph nodes, or the thymus, and further differentiate there. Here are sort of the big three that I would like you to sort of understand. And again, the third one is a group of cytokines. I will give you a couple of examples here in just a second on those. But the first two, one is called erythropoietin, abbreviated EPO. It's made by kidney cells, among other things, and it will drive the creation of red blood cells. Erythropoietin is one way to dope blood. You can give yourself erythropoietin, and it will cause more red blood cells to form. On the therapeutic side, people who are experiencing anemia will be often prescribed EPO in order to boost their red blood cell count. People undergoing chemotherapy, and what happens is by giving them EPO, they get more red blood cells, they tend to feel a little bit better, and so forth. Thrombopoietin, TPO, will drive the formation of megakaryocytes and increase platelet concentration. So people who have issues with blood clotting disorders where they are not able to clot as well, sometimes TPO is prescribed. The last one we're going to call CSFs, colony stimulating factors, and that comes from the phenomena that when you grow these white blood stem cells in the petri dish, you give them these cytokines and you start to see these differentiating colonies. In any case, these guys are the cytokines that will drive specific types of differentiation, so all blood types. So let's look at a chart. I'm just going to show you a rather complex figure in the next one, and you can sort of spot these colony stimulating factors and cytokines. So here's EPO right here. You can see EPO for red blood cell production, so EPO. Here's TPO for platelets, okay? And then CSF, that stands for colony stimulating factor, and there are different kinds. There's GCSF and GM and M, GM. So these are different types of cytokines that will drive differentiation. So again, the big three, erythropoietin for red blood cells, TPO for platelets, and then colony stimulating factors for other types of cell types. So hematopoiesis writ large is the creation of the cellular elements in the blood. We have leukopoiesis, the production of white blood cells, again primarily driven by the CSFs, the colony stimulating factors. Okay, and what we have is that sort of combination of differentiation and migration. Erythropoiesis, the production of red blood cells driven by erythropoietin, EPO. If we have more EPO, more drive to create red blood cells, what we see is an increase in what's called the percentage of volume that is occupied by cells. What we do is we take the blood, we spin it down and pack those red blood cells, and then we look at the volume, and we get a percentage of volume, and that's known as hematocrit. So a hematocrit of 45 means that 45% of the volume of your blood is occupied by red blood cells. So again, if we take a blood sample, and it really doesn't matter how much we have, whatever that volume is, let's say it's 50 microliters, let's say it's 100 microliters. 100 microliters, that would be 100%. If 42 microliters of that 100 are occupied by red blood cells after we spin it down, that gives us a hematocrit of 42. White blood cells usually occupy less than 1%. If we see anything more than 1%, what we call a buffy coat, a light, tiny layer that's just barely visible, that means we probably have elevated white blood cells. So we see somewhere around the order of 42 to 50% by red blood cells, a little higher in men than women. That percent volume occupied by red blood cells is the hematocrit. Now there's a lot going on this slide, and I really don't want to belabor this slide too much, other than to show you down here that the number one population of cells in the white cell population is neutrophils by far. We do see some lymphocytes in there, but over half, just like over half of all the volume of the blood is red blood cells, over half of all of the white blood cells are neutrophils, and that's the only point of this slide. So here's just a sort of a semi-clinical breakdown for you when it comes to hematocrit versus white blood cells in the buffy coat. Over here, here's normal blood. So you can see that females somewhere run around 37 to 47, males 42 to 52. And so here's your hematocrit, the red blood cell packed volume. Here's your buffy coat, the white blood cells, and here's your blood plasma. If we have depressed hematocrit, we would see a lower number for hematocrit. Buffy coat doesn't change, but we do see elevated percentage of plasma in this case. Polycythemia is too many red blood cells. Too many red blood cells would be that you would see a higher hematocrit, low blood plasma, and again, buffy coat is normal. This can happen due to dehydration. It can also happen to disorders that cause elevated levels of EPO. It can also happen because of blood doping. On the other side, if we look at, and again, here's your normal blood, and it's not showing the buffy coat very well here. Here's anemia, here is polycythemia, and here is dehydration. And I don't have a good leukemia one. Leukemia one, you'd see a higher than 1% buffy coat. So now let's switch to red blood cells specifically. We've talked about hematocrit over the last few slides. Let's get into the cell morphology and the shape and the function of red blood cells. Red blood cells are one of the few internal cells that we're actually quite familiar with in our own personal lives because we all cut our cells from time to time. And so it's one of the more frequent events exposing our internal environment to our eyes and our senses. They have this unusual cell morphology. They're relatively unusual in their shape, and that's driven by the cytoskeletal configuration. The cytoskeleton actually drives this biconcave shape. And there are two reasons for this. One is to increase the surface area to facilitate gas exchange, but the other is actually to allow it to be foldable. That is to say, grab, in your own mind, imagine you have one of those paper plates that you have at picnic times. You know how floppy those things are? They're always spilling, and you always got to have more than one or you're going to dump your food on the ground. Well, that's how a red blood cell should behave. It not only has that sort of dinner plate shape, but it also is very foldable to allow it to fit through the narrowest of capillaries. So this is not only for surface area purposes, it's also for perfusion purposes, to allow red blood cells to squeeze into the smallest capillaries that are out there. If we have issues with the blood, one of the ways that we determine those issues is by cell morphology. If we put a red blood cell in a hypertonic media, and hypertonic media means the red blood cell is going to shrivel and lose water, then we start to see this crenation. The cytoskeleton is still there, but what you see is this sort of spiky surface. On the other hand, if we put it in a hypotonic media, that allows water to rush in, and this is a gentle hypotonic media, a little bit of swelling so it doesn't rupture the cells overtly like we did, we would see a loss of that biconcave shape. But we do assess cells based on morphology. And here is a sickled red blood cell. And what happens in sickle cell anemia is that there's a single mutation that triggers, under certain circumstances, the hemoglobin to polymerize. You remember in the previous slide, I mentioned that the foldability is very, very important for red blood cells. A sickled red blood cell cannot fold. Imagine trying to walk through a door where your arms and legs are stuck out as far as they can. Never mind walking, okay? Imagine your arms and legs stuck way out wide. It makes it that much more difficult for you to get through a door, much less a narrow door, okay? And that's what happens in sickle cell. A lot of the symptoms of sickle cell are pain, and that's pain due to ischemia. So we have this sort of system-wide ischemia because all these sickled red blood cells jam up at the entrance of capillaries, leading to deprivation of oxygen and glucose in those tissues. And so those tissues just start screaming for more oxygen and more glucose, cytokines, and you get pain. Again, morphology can provide clues to the presence or absence of disease. If we see cells that are too small, we call them microcytic. Cells that are normal size, normocytic. We can actually assess the volume. This is a typical normal red blood cell, and this is actually on the tip of a pin, like a very sharp, punchboard pin. But red blood cells should have that biconcave shape. They should have the right volume, and they should have the right color. Speaking of color, what drives the color in red blood cells? A molecule known as hemoglobin. And I would like you to understand the components of hemoglobin. Hemoglobin is comprised of 4 separate polypeptides that form a tetramer, 2 alphas and 2 betas. Turns out that neonatal hemoglobin, the hemoglobin that is found in newborns and in preterm infants that are still in utero, actually has a different form of hemoglobin with a higher affinity. Why? Because infants in utero don't breathe, and so they have to have a higher affinity hemoglobin in order to attract oxygen onto their hemoglobin off of the mother's hemoglobin. Interesting story. In any case, 4 polypeptide chains, 2 alphas, 2 betas. Those polypeptide chains stabilize an organic molecule known as heme. It's a carbon molecule. It's not circular. It's actually this sort of cubic looking shape. This is just the diagram. In any case, this is an organic cage for iron. Iron is a sleeping giant, and loose iron in the body is toxic, like you wouldn't believe. I actually did some experiments when I was at the University of Montana early on in my postdoctoral career, where we gave ferrous iron to endothelial cells, and it just absolutely tore them apart. It was actually astonishing how toxic loose iron is. And so we keep iron under lock and key because we need its ability to attract oxygen and release oxygen selectively, but we can't allow iron just to go around on its own. So we keep it inside this thing called a porphyrin ring. There are different types of porphyrin rings out there for different types of hemoglobins. But we keep that iron under lock and key. So we have 4 polypeptide chains, 1, 2, 3, 4, 2 alphas, 2 betas, stabilizing the heme. And the heme is a porphyrin ring with an iron atom at the center. And like I said, there are all different kinds of porphyrins out there. Porphyrins aren't just for hemoglobins. They make wonderful molecular cages, and many chemical engineers are often involved in porphyrin research for caging what are potentially therapeutic molecules that also could be potentially harmful. Since we're getting close to our half-hour time limit on online lectures, I'm going to wrap up with the final 3 slides with this one big mechanism for today, which is hemoglobin and iron transport throughout the body. And so that'll finish it up for today, and then we'll pick it up for the rest of this lecture at the next time. Understand that iron is kept under lock and key. If we tried to inhale iron, or we tried to inject it in our bodies through some other way, we would find incredible levels of toxicity. As I mentioned, when we exposed endothelial cells to native iron, it ripped them apart through free radical production, spontaneous free radical production. And so we absorb it very, very specifically in our diet through active transport. We actually have enzymatic transporters that require B vitamins as cofactors. That's why B vitamins are important nutrients for proper iron absorption. We absorb that iron specifically, and we can regulate it very, very carefully, and then hand it off to the iron carrier in the blood plasma, the protein known as transferrin. Again, we're keeping iron under lock and key so that it can't spontaneously auto-react. As that blood with transferrin in it, that blood plasma, reaches the bone marrow, there are special receptors on the surface of the red blood cell precursors that allow the iron to be transferred from transferrin and sent into the red blood cell and put onto the molecule heme. That's that porphyrin ring and those 4 that porphyrin ring. And so we create heme. It's then conjugated with the proteins to make hemoglobin, and then we finish off blood cells. So when the red blood cell is matured, it contains that hemoglobin. Red blood cells live for about 4 months, around 120 days in the blood. And as those cells get older, they start getting ragged. That's the best way I can put it. They start expressing proteins on the cell surface that they shouldn't be, or there are other sort of markers on the red blood cells that says this red blood cell is getting too old. The spleen has splenocytes, and there are other places in the body that also do this, but the spleen is the major area for red blood cell destruction. They can recognize those red blood cells. It will destroy them in a regulated fashion so that we don't release that iron. So we're going to break those red blood cells down, and we're going to metabolize that hemoglobin into a molecule that has a sort of a golden, off brown-green color called bilirubin. Bilirubin is then metabolized further. Either bilirubin can be excreted by the kidney, and that's the yellowish golden color that you see in urine is by the metabolites of bilirubin or bilirubin itself. But it turns out that bilirubin is not primarily excreted by the kidney. Bilirubin is actually often metabolized by the liver. In case of need for iron, liver does store a lot of iron with a molecule known as ferritin. So we transfer that iron off of the bilirubin onto ferritin, and we have a liver storage pool of excess iron that we keep here, which can be handed off to transferrin in times of need. But most of the bilirubin is actually metabolized and excreted out the bile. We call this the biliary excretion route, and that's what the word bilirubin comes from. So we excrete it in the bile, and most of the bilirubin is excreted in the fecal route. That's what gives feces its brownish color, actually, is the metabolites of bilirubin and bilirubin in the fecal material. That's what gives it its color. So the majority of bilirubin from red blood cell destruction is actually excreted by the biliary route. And so that wraps up part 1 of chapter 16. Make sure you understand these steps here. What are the primary components? How this works? I would never ask you to memorize this pathway entirely, but I would ask you questions about what's going on and where. What are the primary excretion routes of hemoglobin and bilirubin? What would be the molecule that we store iron with in the liver? How do we transport iron and so forth? So it's not a memorization mechanism so much as understand what's going on and where. All right, well, thank you for your time. We're just at 30 minutes. I will see you in part 2 of chapter 16. Talk to you soon.